So under this molecular basis of inheritance, we are first going to discuss about the structure of DNA and how it is packaged inside the nucleus, and then how they found that DNA is the genetic material. So about the experimental studies. And then the most important aspect is the central dogma, that is how the protein is synthesized. So it involves three processes. One is replication, transcription, and translation. So these are the main topics that we are going to cover in this chapter. And then about the genetic code and regulation of gene expression, and finally about the Human Genome Project and DNA fingerprinting. Okay, so today we'll focus on the structure of DNA, structure of the polynucleotide chain, so how it is arranged, and packaging of DNA helix. So these three topics will focus today. So already we discussed about the inheritance, right? So how the characters are transferred from parent to progeny. So we discussed fully about inheritance. And regarding the molecular basis of inheritance, and we also discussed the basic details about the structure of DNA under biomolecules, right? About the structure of nucleic acids. So everything we already discussed. So we'll have a brief discussion about the structure of these nucleotides, and then we'll focus on the packaging of the DNA. Okay. So if you take the nucleic acid, so what are the different types of nucleic acids? We all know it is DNA and RNA. So what is DNA? It is deoxyribonucleic acid and ribonucleic acid. They are the building blocks, right? So if you take the DNA, so here they are made up of the nucleotides. So nucleic acids are commonly DNA and RNA. And in most of the organisms, DNA is the genetic material. So DNA acts as a genetic material in most organisms. But RNA is also a genetic material in some viruses. So they contain RNA as the genetic material. So what is the main role of RNA then? It acts as a messenger. So there are different types of RNA. That is mRNA, which is also called messenger RNA. Then tRNA or transfer RNA. Then rRNA or ribosomal RNA, which is a constituent of the ribosome. So this is the transfer RNA and messenger RNA. So these RNA, mainly they function as a messenger or as an adapter molecule, right? In the case of translation, that is, it carries the amino acid corresponding to the codons which are present in the mRNA. So while studying about the genetic code and translation, we'll see in detail about the function of tRNA. So rRNA are the component of ribosomes. Okay, but if you take DNA, that is the genetic material in most of the organisms. So if you take the structure of DNA, that is the polynucleotides. So what do you mean by polynucleotides? That is, the several nucleotides are added to form a polynucleotide. So this is the structure of polynucleotide. They are the polymers of nucleotides. So first, what is a nucleotide? So we already saw about the structure of bases, right? Nitrogenous bases, then sugar, and phosphate, right? So I hope you're clear with these structures because we already discussed in detail under biomolecules. So this is the structure of a polynucleotide, that is, it has a sugar. If you take DNA, it is deoxyribose sugar, and RNA, it is ribose sugar. And then nitrogenous bases. So these are the nitrogenous bases that are present. C is cytosine, G is guanine, T is thymine, and this is adenine, and they are linked together by means of a phosphate. This phosphodiester bond. Okay, so this is the 1N, which is 3-hydroxyl. So here the 3-hydroxyl is free. But here, this is the 5-N. Here 5-phosphate is at the free end. So all the others, they are linked together by a phosphodiester bond. So these are the sugars, right? So when you take DNA and RNA, they differ in the type of the nitrogenous bases that are present and also the type of the sugar, ribose sugar and deoxyribose sugar. Okay, so these are the basic differences, and this is the structure of a polynucleotide. Okay, so if you take a nitrogenous base, thymine, that is 5-methyl uracil. So you know the structures, right, about the purine and the pyrimidine bases. So if thymine is present only in DNA, thymine is the nitrogenous base which is present only in DNA, but uracil is present only in RNA. So there is main difference with regard to the nitrogenous base. Okay, and purines are adenine and guanine, and pyrimidines are cytosine, thymine, and uracil. So these are the main nitrogenous bases that are present in DNA and RNA. Okay. Then if you take the structure, that is how they are linked. That is, the nitrogenous base is linked to the pentose sugar through a bond that is called the glycosidic linkage. That is an glycosidic linkage to form the structure that is nucleoside. So base which is linked to the sugar. So now if it is linked to the phosphate, then it will become a nucleotide, right? So base plus sugar, it forms a nucleoside, okay, through the N-glycosidic linkage. So a nitrogenous base is linked to, this is the pentose sugar. There is a 5-carbon sugar which may be ribose or deoxyribose. So the nitrogenous base is linked to the pentose sugar through a N-glycosidic linkage to form a nucleoside. Okay. Then in the case of RNA, so what are the type of nucleosides that are present in RNA? Adenosine. So adenosine is adenine plus sugar. Ribose sugar, it is adenosine. Here adenine plus deoxyribose sugar, which is deoxyadenosine, which is present in DNA. So these are the names of nucleosides which are present in RNA and DNA. And guanosine is a nucleoside which is present in RNA. So guanine plus ribose sugar. Here guanine plus deoxyribose sugar, it gives deoxyguanosine. And here cytidine. Cytidine is a nucleoside, right? So bases adenine, thymine, guanine, and cytosine. So cytosine, when it combines with the ribose sugar, it is cytidine, and here with the deoxyribose sugar, it is deoxycytidine. And here the difference is, here it is uridine, and here it is thymidine, that is deoxythymidine. It is present only in DNA. So uracil plus ribose, here thymine plus deoxyribose sugar. So these are the names of the nucleosides which are present in DNA and RNA. Okay. So now in the case of RNA, each nucleotide has an additional hydroxyl group at the second position or 2' position of ribose, that is 2'OH. So these are the structures of ribose and deoxyribose. So what is the difference here? So this is the first carbon atom, second, third, fourth, and fifth carbon atom in a 5-carbon sugar. And here it is 1, 2, 3, 4, and 5. So in the case of ribose sugars, where it has a hydroxyl group at the second position, and here in deoxyribose, it has hydrogen. Okay. So in RNA, each nucleotide has an additional hydroxyl group at the 2' position of the ribose. Okay. The two nucleotides are linked through 3' to 5' phosphodiester bond to form a dinucleotide. So two nucleotides are linked by means of a 3' to 5' phosphodiester bond. So when more nucleotides are linked, it is a polynucleotide. So many nucleotides, they link together to form a polynucleotide. Okay. So who found out the DNA? It is Frederick Mischer. So he was the one in 1869. He identified the DNA and he named it as nuclein. So first he found out the DNA and he named it as nuclein. Later, but the structure of DNA was not elucidated at the time. So later, Watson and Crick, right? James Watson and Francis Crick, they proposed the double helix model of DNA. So the famous double helix model of DNA was proposed by Watson and Crick. So on what basis? So how did they find the double helical structure of DNA? That is based on the X-ray diffraction data produced by Morris Wilkins and Rosalind Franklin. So Morris Wilkins and Rosalind Franklin, they found the X-ray diffraction. So based on the X-ray diffraction data, Watson and Crick, they proposed the structure of DNA as a form of a double helix. So it is the double helix model of DNA. So we'll see in brief about the double helical structure of DNA. So here the DNA is made up of two polynucleotide chains, right? So these are the two polynucleotide chains. They are coiled in a right-handed fashion. So this is main. That is, they are coiled in a right-handed fashion. That is right-handed helix. So the common structure of DNA is the B form, right? So B DNA. And there are different other forms like A DNA and also Z DNA, right? So all these, so this is left-handed. Z DNA is left-handed. This is the common DNA, and there are different forms based on the, that is, they differ in the diameter of the helix and also pitch and the number of base pairs in the per turn. So they differ in the internucleotide spacing. That is how the nucleotides are arranged. Right? So the pitch of the helix in the case of normal B DNA, so pitch is this distance. It is between the one turn to the other turn. So here it is 3.4 nanometer. But here in between the base pairs, it is about 0.34. So here within one turn, if you take one turn from here to here, we can see there are 10 base pairs. So the number of base pairs in each turn is 10. The pitch of the helixes, so the overall distance is 3.4 nanometer. So the distance between a single base pair or the adjacent base pairs is 0.34 nanometer. Okay. So if you take the backbone, so this is the backbone. The blue colored structure, it shows the backbone of the DNA, right? So here it is made up of sugar and phosphate. So sugar and phosphate mainly, they form the backbone of the DNA, right? So what about these lines? So these are the nitrogenous bases which are present inside. So the bases project inside. So here this yellow, it represents the adenine, and green, it represents the thymine, and this orange one, it represents the guanine, and this red, it represents the cytosine. So adenine always pass with thymine, right? Adenine will always pass with thymine by a double bond, and guanine will always pair with cytosine by a triple bond. So here there will be 3 hydrogen bonds. So what are the types of bonds that exist in a nucleotide? That is, they have the hydrogen bond and phosphodiester bond, and also they have the glycosidic linkage. So between sugar and the base, it has a glycosidic linkage, and in between the nitrogenous bases, we have the hydrogen bonding and phosphodiester bond, which links the nucleoside, right? So nucleoside plus phosphate forms a nucleotide. Okay, so 3 types of bond exist here. So if you take the structure of DNA, the 2 chains have anti-parallel polarity. So what do you mean by anti-parallel polarity? So if you take this, so this is one chain and this is the other chain, right? 2 polynucleotide chains. This is the one, this is the other one. So they are of anti-parallel. That is, one chain is in the 3 dash. It starts from 3 dash and it goes like this. It's in the form of coil form, and this is the 5 dash. And so 3 dash to 5 dash. If you take the other chain, here it is 5 dash to 3 dash, right? So they are of anti-parallel. So one chain has the polarity of 5 dash to 3 dash, whereas the other has the polarity of 3 dash to 5 dash. And the bases in the two strands, they are paired through hydrogen bonds forming base pairs. Okay, so here between the base pairs, that's what we saw, they are linked by hydrogen bonds between the adenine and thymine and between guanine and cytosine. Okay, so purine comes opposite to a pyrimidine. So this generates a uniform distance between the two strands. So adenine and thymine. So adenine is a purine, right? And thymine is a pyrimidine. So there always occurs a bonding between purine and the pyrimidine. So whenever a purine is present here, and pyrimidine will be present here in the next nucleotide chain. So their purine comes opposite to a pyrimidine. So as a result of it, that uniform distance is maintained. So guanine is a purine base and cytosine is a pyrimidine base. So purine always binds with the pyrimidine. So it helps to, if two purines actually they pair together, purine is a two-membered ring, right? So there will be no spacing. But if pyrimidines are present, there will be more space. So it occurs in such a way that there always exists a bonding between a purine and a pyrimidine. That too only between adenine and thymine and between guanine and cytosine. So this is more important. Okay. So purine comes opposite to a pyrimidine and it helps to create a uniform distance between the two strands. So there are two groups. One is called the major group, the other one is called the minor group. So these are the base pairs and sugar and phosphate backbone is in this blue structure. Okay. So what is Erwin-Chargaff rule? That is, it shows us the pairing between the nucleotides in the double helical structure of DNA. So in the DNA, actually the adenine is equal to thymine always, and guanine is always equal to cytosine. We know that there occurs a pairing between adenine and thymine. So always the amount of adenine will be equal to that of thymine and the amount of guanine will be always equal to cytosine. And also A plus G, that is purine, will be equal to pyrimidine, right? That is A plus G will be equal to T plus C. Adenine plus guanine is equal to thymine plus cytosine, or A plus G by T plus C is equal to 1. So these are the basic rules, right? That is Erwin-Chargaff rule. But if you take the content of A plus T by G plus C, they may vary from one species to another. They vary from one species to another, but it is constant for a given species. For example, in some cases, they will be 80 rich. That is, the base ratio will be then greater than 1 if they are rich in 80 sequences. But in some cases, GC rich, then the base ratio will be less than 1. Okay, so it depends on the ratio. That is, it varies from species to species, the content of AT and GC. So for example, if you take the, what do you mean by denaturation of DNA? That is, if you expose it to a high temperature, this double helical DNA will separate to form a single stranded form, right? So if the DNA is rich in GC, that is, the bonding will be more stronger because of the triple bond, right? So there the melting temperature is high in the case of DNA with GC rich base pairs. Okay, so it varies from species to species. And if you take pi X174, it is a bacteriophage. It is a bacteriophage which has a nucleotide of about 5,386 nucleotides. And bacteriophage lambda, it has about 48,502 base pairs. And E. coli has about 4.6 into 10 power 6 base pair. But the haploid content of human DNA is 3.3 into 10 power 9 base pair. So it varies from species to species. That is the number of nucleotides. Okay. So how will you calculate the length of the DNA? So length of the DNA is equal to number of base pairs into distance between two adjacent base pairs. For example, if you take human, the number of base pairs that are present is about 6.6 into 10 power 9. So this is the diploid number, right? So number of base pairs that are present. So if you want to calculate the length of DNA, so it is the number of base pairs into distance. So distance, we saw that it is 0.34 nanometer, right, between the adjacent base pairs. So 0.34 nanometer, and it can be written as 0.34 into 10 power minus 9 meter. Okay, so length of the DNA is approximately equal to 2.2 meters, that is human DNA. And in the case of E. coli, the length of the DNA is equal to 1.36 mm, which can be written as 1.36 into 10 power minus 3 meters. So millimeter, when it is expressed in meters, it is 1.36 into 10 power minus 3 meter. So the total number of base pairs is equal to length of the DNA just divided by the number of base pairs, which gives us that is 4 into 10 power 6 base pairs. So number of base pairs is equal to 4 into 10 power 6 base pairs in the case of E. coli. So here the length of the DNA is 2.2 meters. So actually, if you take the length of the DNA, it is it's in the highly lengthy, right? 2.2 meter, but it has to fit into the nucleus. So how that is happening? So it has to be in the form of coiled form, and it has to be packaged, right? So it cannot be straight because the length itself is 2.2 meters. So how the packaging of DNA is happening? So in the case of prokaryotes, so if you take prokaryotes, particularly in E. coli, the DNA is not scattered throughout the cell. So the DNA is not scattered, but it is present in the coil form. So even if there is no definite, so this is the structure of a prokaryotic cell, right? Capsule, cell wall, plasma membrane, cytoplasm. And if you take the nuclear material, so it is present in the form of a nucleoid, right? So it is not random. So it is present in a particular position. So how it is happening? Because the DNA is negatively charged. Why the DNA is negatively charged? Due to the presence of phosphate, right? Due to the presence of phosphate, the DNA is negatively charged. So as it is negatively charged, it is held with some positively charged proteins. So it is present in the particular position because of the binding between these phosphates and the positively charged proteins, and they form a structure that is called the nucleoid. So it is present in the center, right? So it is not present throughout the cell. And if you take the eukaryotes, there is a set of positively charged basic proteins which are called histones. So these are present only in eukaryotes. So these are the specific feature in eukaryotes, right? So here in eukaryotes, there's a set of positively charged basic proteins called histones, and they are rich in basic amino acids like lysine and arginine. So these are all positively charged amino acids, lysine and arginine. So this amino acids, they bind with the negatively charged DNA. So DNA, we saw it as negatively charged due to the presence of phosphate. So they form a structure like this. This is the core of the histone molecule, and the DNA, it wraps around the core. And that is histones. Actually, it exists in the 5 forms: H1, H2A, H2B, H3, and H4. So they exist in 5 forms, and that too they exist in pairs. So these 4, that is H2A, H2B, H3, and H4 pairs, they form, that is 8 histones, right? They form an histone octamer. So that is the center part. They form this green color one, it represents the histone octamer. That is 8 histones, they'll form an octamer. And this is H1 histone. So this H1 histone, it alone exists as a separate entity. So this negatively charged DNA is wrap around histone octamer to form a structure that is called nucleosome. So they together, they form a structure that is called a nucleosome. So this is how the DNA is wrapped and it is placed inside the nucleus. Okay, so they're not present in a random form because of the binding between the negatively charged phosphate that is present in the DNA and the proteins that are called the histones, which are rich in basic amino acids. So the negatively charged DNA is wrapped around the histone octamer to form a nucleosome. Okay. So for example, if you take a typical nucleosome, so this is the structure of a nucleosome. It contains about 200 base pairs. Okay. This is 200 base pairs. That is single nucleosome, which has the octamer, histone octamer, and the DNA wrapped around it. That is called the nucleosome. Therefore, the total number of nucleosomes in a human DNA is, so these are the total number of base pairs. So these are the base pairs which are present in a single nucleosome, right? So if you calculate, this is equal to 3.3 into 10%, which is equal to the total number of nucleosomes that are present in a human DNA. Okay. So are you able to follow this calculation? That is, how are we calculating the length of the DNA, and here how the total number of nucleosomes we are calculating. Are you clear with this part? Any doubts? So clear, right? Okay. So in the case of eukaryotes, the nucleosomes constitute the repeating unit to form a structure that is called the chromatin. So the nucleosomes constitute the repeating unit to form the structure that is called the chromatin, right? So this will be how they are arranged. They are arranged like a beads on the string. So these are like a beads, and this is a string. So this is one nucleosome, that is histone core, which is wrapped with this blue color. It represents the DNA, right? So these nucleosomes, they constitute the repeating unit to form chromatin. Chromatin is the thread-like stain bodies. So these chromatins, actually here, if you see the structure, this blue is the DNA, and this yellow ball-like structure, it is a nucleosome. So this is the histone core, and this DNA is called the linker DNA. That is DNA which links the two nucleosomes. So in between the two nucleosomes, they are linked by a DNA. This is called the linker DNA. Okay, so they are arranged in the form of a bead. So this is like a bead, and this is another bead, that is another nucleosome. So each nucleosome, it contains 200 base pairs, and they are linked by the DNA which is called the linker DNA. So the nucleosomes constitute the repeating unit to form a chromatin. So this is the chromatin, and it is the thread-like stain bodies. Okay, so the nucleosomes in chromatin are equivalent to beads on the string. Okay, so the chromatin is packaged to form chromatin fibers, and they coil and condense at the metaphase stage to form chromosomes. So they form the structures that are called the chromosomes. Okay, so the chromatin is packaged, coiled, and they condense to form chromosomes. But this packaging, it requires the presence of non-histone chromosomal protein, that is also called NHC, that is non-histone chromosomal proteins. So packaging of chromatin, it requires the proteins. And in the case of eukaryotes, the chromatin has two forms, that is in euchromatin and heterochromatin. So what is the role of euchromatin and heterochromatin? Euchromatin is a loosely packed chromatin, and it is a transcriptionally active region of the chromatin. So this is the most active part of the chromatin, but heterochromatin is the inactive region of the chromatins. So the chromatin has two forms, that it exists in two forms. One is euchromatin, the other one is heterochromatin. One is loosely packed, and here it is densely packed. That is the active form of chromatin, and this is the inactive form of chromatin. And it stains light with staining agents, but here it stains dark. So these are the differences between euchromatin and heterochromatin. Clear? So this is about the packaging. So the DNA cannot exist as such because the length of the DNA is about 2.2 meters, which cannot exist as such. So that occurs, and they exist in the highly condensed or the coil form. So that is the packaging of the DNA helix inside the nucleus, which is with the help of the proteins. In the case of eukaryotes, they are called histones. Those proteins are called histones. So there occurs a binding between the negatively charged DNA and the positively charged proteins. That is amino acids which are present in the proteins. So as a result, the DNA exists in the highly condensed or the coiled form inside the nucleus. Okay, so any doubts in this? So with this, complete almost all these we discussed right under biomolecules itself. So we know that there are different forms of DNA like aDNA, and you want me to repeat those, you know, right? About the different forms of DNA, and we also studied about the different bonds that are present. Yes. So are you clear with that? So there are 3 other types of DNA, right? Okay, so we'll discuss few questions then. So all these things, I hope you're clear with the structure of DNA. And tomorrow we'll discuss about the experiments to prove that DNA is a genetic material and other experimental things. So today we'll discuss few questions which are related to this structure of DNA. Okay, so here you have to be clear with the Chargaff rules. Maybe some questions they may ask relating to the Chargaff rule. So that is the content of adenine is equal to thymine and cytosine is equal to guanine, and about the ratio, right? Only those things, and other questions maybe only direct questions will ask. So you have to be clear with the double helical structure of DNA. What is the pitch, and what about the distance between the nucleotide? So those type of things you have to be clear. And also what is a nucleoside, what is a nucleotide, and you have to be clear with the names, that is adenosine, guanosine, and then cytidine, thymidine. So about the names of the nucleoside and the nucleotide. So these are the things that you have to focus in this topic. Okay. So, and there are different types, that is A, B, C DNA and Z DNA. Among these are A, B, C or right-handed DNA, and Z DNA is a left-handed DNA. Okay, so we'll discuss few questions. So hydrogen bonds occur between which of the following constituents of DNA? Is it between sugar and the base, phosphate and base, complementary bases, phosphate and sugar? So where the hydrogen bonds occur between any one? Yes, C is the right answer. So the hydrogen bond exists between the complementary bases, right? So we saw that between adenine and thymine and guanine and cytosine. So here there exists the presence of hydrogen bonding, but sugar and base, they are linked by the bond, that is glycosidic, N-glycosidic bond. So the other type of bonds are glycosidic bond and phosphodiester bond. Okay, and phosphodiester bond. But hydrogen bond exists between the complementary bases. So C is the right answer. So in one strand of DNA, the sequence of bases is this. Then the complementary sequence of bases on the other side of the strand would be. So what will be the correct answer here? So you have it in mind always that adenine pairs with thymine and guanine pairs with cytosine. So based on that, you write the complementary sequence of these. Okay. Is B is the right answer. Yeah. So yeah, you able to follow Afia, Himanshu? So B is the right answer, right? Because if you take the sequence, that is first A, T, G, G, C, 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 T, T. So complementary basis for adenine, it is thymine, and here adenine, thymine, thymine. So for thymine, it is adenine. For guanine, it is cytosine. Cytosine. For cytosine, it is guanine, guanine, adenine. So the correct answer is B. So you're clear with the complementary bases, right? Yeah. What is the distance between two base pairs in the double helix model of DNA? Between two base pairs, between one base pair and the next base pair, what is the distance? Whether it is 0.34 nanometer, 34 nanometer, 24 nanometer, 0.034 nanometers. So which is the right answer? What is the distance between two base pairs in the double helical model of DNA? Yes, A is the right answer. That is 0.34 nanometer is the right answer. The pitch of the helix is 3.4 nanometer and the number of base pairs per turn is 10. So the distance between two base pairs is 0.34. So A is the right answer. The difference in A, B, and Z forms of DNA lie in base sequences, parallelism of chains, hydrogen bonding, internucleotide distances. So in what way the different forms of DNA differ? Actually, the B DNA is the form that we studied, right? What we saw, the double helical structure is the structure of B DNA. So the difference in A, B, and Z forms of DNA, they lie in on what basis they differ. So the correct answer is D. Actually, that is they differ in the internucleotide distances. For example, if you take ADNA, the distance between the base pairs is about 2.56 Armstrong. But if you take BDNA, it is 3.4. So they differ in the internucleotide distance and also the diameter. They differ in the diameter and also the number of base pairs per turn. So everything there is a different, but there's no change in the hydrogen bonding or parallelism. That is, both will be in the anti-parallel. So those factors, they won't change, but there occurs a difference in the internucleotide distances, right, between A, B, and Z form of DNA. But only the Z DNA is left-handed form, and these are all right-handed DNA. So they also differ in the number of base pairs per turn, diameter of the helix, and the distance between the base pairs. Okay, and also these DNA, some DNA, they exist in the palindromic form. What do you mean by palindromic DNA? That is, if they read from the both the 5' to 3' and they will be same. For example, C, G, D. So this is in this direction, and here it will be G, C, A, T. So if you read from the opposite ends, the sequence will be same. That is the palindromic DNA. So palindrome means generally when you read from front to back or from back to front, it will be the same, right? So this is the palindromic DNA. So if in a DNA molecule, cytosine is 18%, what is the percentage of adenine? So what will be the answer for this question? Can you calculate? If in a DNA molecule, cytosine is 18%, what is the percentage of adenine? So as per the Chargaff rule, the cytosine will be equal to guanine because there occurs a base pairing between cytosine and guanine. So cytosine is 18%, then guanine will be 18%, right? So if you add these two, what is the value of C plus G? So what is the value of C plus G? It will be 28, 30, 36, right? So then what will be the percentage of A plus T? So the remaining will be A plus T, right? So what will be the answer? Yes, B is the answer. So A plus D will be 64. Because together they form 100. So A plus D is 64. Then what they asked is the percentage of adenine. So the adenine is half of it. So 32% of adenine, and thymine will be 32. So are you clear with this? So based on the Chargaff rule, that is cytosine is equal to guanine. So C18 means G also will be 18. So C plus G content is 36 and remaining will be A plus T. So that is 64%. So A is 32 and T is 32. So the answer is B. Okay. So if they ask any type of question like this, so you should be able to do it. Nucleic acids are polymers of nucleotides, nucleosides, amino acids, nucleoproteins. So nucleic acids are polymers of, yes, A is the right answer. That is nucleotides. So clear? Clear with this, right? So we'll discuss few more questions. The double helix model of DNA, which was proposed by Watson and Crick, was of cDNA, bDNA, ddNA, zDNA. So which is the right answer? The double helix model of DNA, which was proposed by Watson and Crick, was of cDNA, bDNA, ddNA, then zDNA. So which is the right answer? Is B is the right answer. That is bDNA. So bDNA is the right answer. Okay. And if there are 10,000 nitrogenous base pairs, if there are 10,000 nitrogenous base pairs in a DNA, how many nucleotides are there? There are 10,000 nitrogenous base pairs are there. In a DNA, how many nucleotides are there? Option E, 500, 10,000, 20,000, then 40,000. So what is the answer? If there are 10,000 nitrogenous base pairs in a DNA, then how many nucleotides are there? So what is the question? If you take base pairs, it is given. No, it is not A. What they meant is, for example, in a double helix. So this is the helical structure. Imagine. So here they told about the nitrogenous base pairs. The base pair is, that is here if there is A and T. So this is one base pair, right? So like this, there are 10,000 base pairs. So the question is, how many nucleotides? So nucleotides will be in a single chain. So this is one nucleotide. This is linked to the other one. So base pairs or 10,000 means if you take the nucleotide as it is, it will be double. Can you understand? So the answer will be 20,000. So this two, they form a base pair. So they individually, if you calculate, it is 2, right? If it is a pair, this is 1. But if you treat it as an individual nucleotide, these are 2. So 10,000 means it will be 20,000. Yeah. That is, the question is, if there are 10,000 nitrogenous base pair, so it is given as base pair. So this is one base pair. So this base pair, it has 2 nucleotide, right? A and T, it has 2 nucleotides here. Here there will be sugar and the phosphate. So if you take this one base pair, it has 2 nucleotides. So if you take 10,000 base pairs, it will have 20,000 nucleotides. Clear now? Okay. Double helix model of DNA is proposed by Watson and Crick, Skleden and Schwann, Singer and Nicholson, Kornberg. So the correct answer is A, Watson and Crick. Backbone in structure of DNA molecule is made up of a pentose sugar and phosphate, hexose sugar and phosphate, purine and pyrimidine sugar and phosphate. So which is the right option? Is it is A, that is pentose sugar and phosphate. Backbone in structure of DNA molecule is made up of pentose sugar and phosphate. A is the right option, and it is not hexose sugar, and it is not purine and pyrimidine. Purine and pyrimidine, they form only the middle, not the backbone, right? So pentose sugar and phosphate is the right answer. Substance common in DNA and RNA. So what are the substances that are present in both DNA and RNA? It's a hexose sugar, histamine, thymine, phosphate groups. The substance common in DNA and RNA are hexose sugar, histamine, thymine, and phosphate groups. Yes, D is the right answer. Right. So Himanshu, are you able to follow? You can give your answers. Then nucleotide is option E, nitrogen base, pentose sugar and phosphoric acid. Option B, nitrogen, hexose sugar and phosphoric acid. Option C, nitrogen base, pentose sugar. Option D, nitrogen base, triose and phosphoric acid. The nucleotide is, A is the right answer. That is, it has nitrogen base, pentose sugar and phosphoric acid. DNA differs from RNA in only sugar, nitrogenous base only, and option 3, nitrogenous base and sugar, and none. DNA differs from RNA in, yes, C is the right answer. That is, they differ in both nitrogenous base and the sugar. That is, thymine. Here, uracil is present in RNA. There, sugar, deoxyribose sugar, and in case of RNA, it is ribose sugar. So the unit of nucleic acids are phosphoric acid, nitrogenous bases, pentose sugar, and nucleotides. Units of nucleic acids are phosphoric acid, nitrogenous base, pentose sugar, and nucleotide. Yeah, nucleotide is the right answer. Which element is not found in nitrogenous base? Nitrogen, hydrogen, carbon, phosphorus. So which element is not found in nitrogenous base? Nitrogen, hydrogen, carbon, and phosphorus. So you think of the structure of nitrogenous base alone. That is structure of adenine, guanine, then cytosine and thymine and uracil. So which of the element is not found? So nitrogen, hydrogen, carbon, and phosphorus. So here again D is the right answer. Yeah, D is the right answer because phosphorus is actually not found in the nitrogenous base only. It helps in the linking, right? Only in the formation of phosphodiester form bond it is involved, but it is actually not a constituent of a nitrogenous base. But nitrogen, hydrogen, and carbon are part of the nitrogenous base. So D is the right answer. Then DNA was first discovered by Mixture, Robert Brown, Fleming, Watson, and Crick. DNA was first discovered by Mixture, Robert Brown, Fleming, Watson, and Crick. Yes, A is the right answer. Mixture. DNA is not present in mitochondria, chloroplast, bacteriophage, and TMV. So what do you mean by TMV? That is tobacco mosaic virus. So DNA is not present in mitochondria, chloroplast, bacteriophage, and TMV. Yeah, D is the right answer because viruses, the TMV contain only RNA and not DNA. But mitochondria and chloroplast, they have their DNA. That is mitochondrial DNA and chloroplast DNA. So D is the right answer. That is TMV. The nucleic acid contains thymine or methylated uracil. Then it should be DNA, RNA, either DNA or RNA of bacteria. The nucleic acid contain thymine. So what it should be? Thymine is also called methylated uracil. Then it should be a DNA, RNA, or either DNA or RNA of bacteria. Yes, A is the right answer. That is, it is a DNA. Thymine is present, it is a DNA. Then prokaryotic genetic system contains DNA and histones, RNA and histones, either DNA or histones. Then option D, DNA but no histones. Prokaryotic genetic system contains. Yeah, D is the right answer. Because prokaryotes, they have DNA but no histones. Histones are present only in eukaryotes. The nitrogen base together with pentose sugar and phosphate, they form the building block unit of nucleic acid is nucleoside, polypeptide, nucleotide, and amino acid. Nitrogen base together with pentose sugar and phosphate. Yes, they form a nucleotide. C is the right answer. One of the characteristics of DNA is uracil, deoxyribose sugar, then single strandedness, ability to synthesize protein. One of the characteristics of DNA is uracil, deoxyribose sugar, then single strandedness and ability of protein synthesis. So which is the right answer? Yes, B, because they have the deoxyribose sugar. So B is the right answer. Which of the following is not a pyrimidine nitrogenous base? Thymine, cytosine, guanine, and uracil. Thymine, cytosine, guanine, and uracil. Which of the following is not a pyrimidine nitrogenous base? Yeah, C is the right answer. It is guanine. Guanine alone is a purine. All the others are pyrimidine. The purine and pyrimidine parts of complementary strands of DNA are held together by hydrogen bonds, oxygen bonds, carbon bonds, nitrogen bonds. Yes, A is the right answer. That is hydrogen bonds. The purine and pyrimidine pairs of complementary strands are held together by hydrogen bonds. The number of hydrogen bonds between guanine and cytosine are 1, 2, 3, and 4. The number of hydrogen bonds between guanine and cytosine are 1, 2, 3, and 4. Between guanine and cytosine, yeah, C, because 3 hydrogen bonds. Which purine and pyrimidine bases are paired together by hydrogen bonds in DNA? AC and GT, GC and AT, GA and TC, none of the above. Yes, B is the right answer. That is the recursion linkage between GC and AT. What is the nature of the two strands of DNA duplets? They are identical and complementary, anti-parallel and complementary, dissimilar and non-complementary, anti-parallel and non-complementary. What is the nature of two strands of a DNA duplex? Yeah, they are anti-parallel and complementary. That is, two strands will be anti-parallel. One in the 5' to 3', the other is the 3' to 5', and they show complementary. That is, A is complementary to T and G with C. So the answer is B, that is anti-parallel and complementary. On an average, how many purine nitrogen bases are present in a single coil of DNA? That is, how many nitrogenous bases are present in single coil? 4, 5, 10, and uncertain. How many nitrogen bases are present in a single coil? That is, in one turn of P helix. So how many base pairs are present? Yes, 10. The Chargaff's rule is given as, that is, purine is not equal to pyrimidine. A plus G is equal to T plus C, or A plus U is equal to G plus C, and A plus G by G plus C is a constant. So which is given by the Chargaff's rule. The Chargaff's rule is given as purine is not equal to pyrimidine. So that is a wrong statement. So the correct statement is, in the case of Chargaff rule, A plus G, that is purine, is equal to pyrimidine. So B is the right option, right? So the purine is equal to pyrimidine. So B is the right answer. A plus G is equal to T plus C. Then in RNA, nucleotides are bonded by hydrogen bonds, phosphodiester bonds, ionic bonds, and salt linkage. The nucleotides are bonded by hydrogen bonds, phosphodiester bonds, ionic bonds, and salt linkage. B is the right answer. That is phosphodiester bonds. It links the nucleotides. A nucleoside differs from a nucleotide in not having phosphate and sugar and nitrogen base. A nucleoside differs from a nucleotide in not having phosphate and sugar and nitrogen base. Yes, A is the right answer. So the main difference between nucleoside and nucleotide is the presence of phosphate. So the nucleoside with phosphate, it forms a nucleotide. Okay, so it forms a nucleotide. Wilkins X-ray diffraction showed the diameter of the DNA helix is 10 Armstrong, 20 Armstrong, 30 Armstrong, and 40 Armstrong. It is the diameter of the normal bDNA. So what is the diameter of the normal bDNA? 10 Armstrong, 20 Armstrong, 30, and 40 Armstrong. 10 Armstrong, 20, 30, and 40. So which is the correct answer? It is the diameter of the helix in the case of bDNA. Yes, B is the right answer. That is 20 Armstrong is the right answer. Diameter. Okay. Similarity in DNA and RNA. Both are polymer of nucleotides. Both have similar pyrimidine. Both have similar sugar. Both are genetic material. So among this, which is the similar between DNA and RNA? Both are polymer of nucleotides. Both have similar pyrimidine. Both have similar sugar. Both are genetic material. Yeah, A is the right answer. That is, both are polymer of nucleotides. But the other three are wrong because they don't have a similar pyrimidine. They differ in their pyrimidine. Both have similar sugar is also a wrong statement because they have two different types of sugars. And both are genetic material. But most of the organisms, DNA is the genetic material. Here only in some viruses, RNA it acts as a genetic material. Okay, so first is the right, A is the right answer. The length of one loop of bDNA is 3.4 nanometer, 0.34 nanometer, 20 nanometer, 10 nanometer. The length of one loop of bDNA is one loop of bDNA. One loop in the sense that is one turn, right? Whether it is 3.4 nanometer or 0.34 nanometer, 20 nanometer, 10 nanometer. No, the question is the length of one loop of DNA. That is the distance between the one. So here there will be a turn, right? Between one turn. That is called the one loop of bDNA. So what is that length? So that is actually 3.4. But here this is between two base pairs. Okay, between two base pairs. And see, this 20 Armstrong is actually the diameter. So the correct answer is option A. So are you clear? The length of one loop of bDNA is actually 3.4 nanometer and not 20. 20 is the diameter of the DNA helix. Okay, so is it clear? The extranuclear DNA is found in option A, lysosome and chloroplast. Option B, chloroplast and mitochondria. Option 3, mitochondria and lysosome. And option 4, Golgi and endoplasmic reticulum. So the extranuclear DNA is found in is B is the right answer. That is chloroplast and mitochondria. So B is the right answer. So I hope you are clear with the structure, right? About the structure of DNA. So any type of questions you must be able to answer. So the only problem oriented question here is that is they may give any content, that is A or C or G or T, and they may ask you to find out the percentage of the other nitrogenous bases that are present. So that is the only problem oriented. So other than that, you may get only the direct questions. So you be clear with the nitrogenous bases and what is a nucleoside, nucleotide, and about the double helical structure of DNA and how the DNA is packaged in the nucleus. That is the structure of histones. So what are histones? So only these are the main factors that will come under the structure of DNA. Okay, so you're clear with it. So tomorrow we'll continue with the experiments. That is how they prove that DNA is the genetic material. So we'll continue in the next class. Okay, so no doubts, right?